Okay, so for the next part, now that we know about the colorful past of CRISPR and the fact that it cuts DNA and exists in bacteria, we're going to look at how we can actually use it experimentally and in the lab. So I'm going to be talking about this along with Gittes. I am also a PhD student at Imperial, and I'm working in the Center of Synthetic Biology under I'm co-supervised by Rodrigo Ledesma Amaro and Professor Tom Ellis. So let's get stuck into it. Experimental stuff.
So there are many types of CRISPR systems, but the ones that are most common are Cas9, Cas12a, and for RNA, it's Cas13a and Cas13b. So what's different about these systems? Cas9 cuts DNA, produces blunt cuts, while Cas12a produces sticky cuts. It means that when Cas9 cuts the DNA, it just leaves like a straight gap, like straight cut, while in Cas12a, it produces overhangs. Cas9a has a G-rich PAM sequence, and Cas12a has a T-rich PAM sequence. And Cas9a cuts the DNA kind of closer to the PAM sequence compared to the Cas12a. And because all of these, and Cas12a is smaller than Cas9, it's better to use for knock-ins. And Cas13a and Cas13b is useful for RNA.
Yeah, so now that there are, like it is explained, there's a lot of natural variety in CRISPR already. You have these different kind of cutting mechanisms, different PAM sequences. That's only what exists in nature. There's even more variety when we look at what we've done with it now. There have been a lot of experiments, like designs using CRISPR. So for example, we can now use CRISPR for repression. So all of these things that we were talking to you before about was about cutting the DNA. Whereas now we also have functions where we can repress gene activation. So for this, they take a catalytically dead Cas9, which means that it doesn't cut the DNA, but it will find your target based on your PAM. And then it is fused to a repression domain. And so it's able to sit onto a specific region, which would be the promoter. And using these repressing domains, and due to like the fact that it's actually sat on the DNA, it will not allow for any transcription. So that's one extra use of CRISPR. You can use it as a repression transcription factor. You can also do the opposite of this and use it as an activation transcription factor. And in these cases, there have been edits to change the CRISPR, the Cas9 or the nuclease, to have activation domains fused to it. So in strategy 2. Or it's been fused to a scaffold, which allows for activator peptides to then be recruited. Thirdly of all, it's also been modified to be epigenetically useful. So in this case, the Cas9 is able to form complexes at spaces where you want epigenetic modifications, and there also is fusions sometimes to epigenetic modifiers. So these new variants of Cas proteins allows for a huge broad range. And on top of what Gittes already mentioned of all these different functionalities of CRISPR, whether it's blunt end or whether it's overhangs, it allows the user of CRISPR to have a big library of tools at their disposal. Generally, when you want to express the CRISPR system in a cell, you're going to need at least 2 elements. And these first 2 elements is something. The first one is the Cas protein. So this would be the nuclease. This has to be expressed under a promoter, be it inducible or constitutive. And then this needs to be transformed in the cell. The second very important element is this guide RNA. And the guide RNA, again, is under a promoter, and this is going to be inserted into the cell, and then it will be expressed. Once these are together, they will form this complex, which will then go and seek out your target to cut it, repress it, activate it, what have you. If we look at a traditional Cas9 enzyme, what happens is it will then find your target region and it will generate this double-stranded break. So your DNA is kind of like cut. And usually that is enough to already generate some knockouts. So you can, using non-homologous end joining, it can generate indels. And the reason that is because non-homologous end joining is one of the mechanisms of repair inside the cell. This mechanism doesn't need any template. It just takes the broken bits and sticks them back together. However, this system is really error prone, which means that it's easy to use for knockouts because you can generate indels and all of that. The big plus of the system is that it's highly efficient and it goes quite quickly. There is another system that we can leverage with CRISPR systems, and that's the homology-directed repair pathway. And in this case, you do need a template. So it doesn't just wed together the 2 bits of DNA. This system is more precise because you're giving it exact templates. You can add something in. It doesn't only knock out. You can put in a new gene there in the middle. You can put the precise edits, or you can put a marker in. And how it does this is because it has these homology regions. So flanking the bit that you want to add in, these homology regions are the same to existing DNA. So when it is repairing it, it's able to recognize these bits and then bring in the bit in the middle as well. So these are the 2 systems. But if you're using homology-directed repair, you also need a 3rd element on top of your nuclease and your guide RNA. You also need a donor DNA, which has these recombination arms. So these will be the same as the regions flanking the area that you want to edit. And between this, you can have a knock-in. So if you want to add in a gene, or an edit if you wanted to change something about the gene that you're targeting, or perhaps just a marker so you can see that it's been put in. I mean, you can even have nothing at all, and it's just increases the efficiency. So these are the 3 elements. Once you get this donor DNA in, what happens is once you generate this double-strand break, like we saw previously, these recombination arms will allow for the bit that you want to go in. So this is much more precise. So the 3 elements that are really important when we're talking about basic CRISPR systems in a cell is the Cas protein, so the nuclease, the guide RNA, and the donor DNA. Now, the ways to get this into the cell are very, very many. There's loads of different kits, different systems, different papers out there. And I mean, it depends on which one is useful, depends on which organism you're going to use, depends on what strength that you want to use. There's a lot of variety in there. And so I can't really show you all of that today. What I will do with you today is go through an example of one that we use here in the Ellis lab, where we have a single guide RNA and Cas9 expression tools for yeast, Saccharomyces cerevisiae. And like previously, it has the 3 elements. The first element being your nuclease protein. So there's a linearized Cas9 guide RNA expression vector. The second is your guide RNA. And the third is the donor DNA, which is just the one which has the recombination or the thing that you want to edit in. If you want to go through this more in detail, there's a link to the Benchling blog, which has all the protocols attached to it, and all the plasmids are already in iGEM. But like I said, this is the one that I've been using. It's the one I'm familiar with, but there are so many different ones like this. So look into what would be the most suitable for your cell type and for your application. In this case, so the first element here is your nuclease. In this kind of toolkit, there is a plasmid that already has your nuclease under some promoters. There's a different variety, so there's 2 different promoters. You can choose which one you want to use. I think they're both constitutive though. And then this plasmid also has a selection marker, which is uracil. And then it has these homology arms. And these homology arms will allow the guide RNA to join this plasmid. But I'll explain that in a bit. So just know that the first plasmid is with your Cas protein. And then you have to linearize it to transform it into the yeast. So to do that, we just do a BSMB1 digestion for a long one for 4 hours or more at 37 degrees. And then we gel purify it to make sure that all we have is the plasmid that's cut, the linearized plasmid. And you just use a simple DNA purification kit or whatever system that your lab is using. The second element that I'm going to talk to you about is the guide RNA part of it. And for the guide RNA part of it, we want to design our own target. So the way that we do it in our lab is that we order 2 oligos that have our target, which would be designed in silico. That will be the next part of this tutorial. There's the part 3 is about how to design the target. But once you have designed it, you get your target, usually around 20 base pairs, and then it will have these overhangs attached to it that we've designed for the rest of the kit. So with these primers, first you phosphorylate them, and then you anneal them. And this allows these 2 oligos, so we order the target and the complement reverse of the target with these overhangs, so that it creates a double-stranded DNA. And this double-stranded DNA is something that's going to slot into the next a plasmid. So once we get this target, we combine it with another plasmid, which is part of the toolkit. And this plasmid already contains the guide RNA scaffold, which is what is necessary for it to bind to the nuclease, and a promoter so that it's expressing the guide RNA. It also includes some ribozymes and things to ensure efficient expression. Like I said, that is dependent on each toolkit. You're going to find variations in that. Once you get that together, using Golden Gate assembly with BSMB1 type 2S restriction enzyme, we remove a GFP, which allows us to select for it, and in its place, we slot in the target, which is the guide RNA. I've included here the protocol for Golden Gate, but you can find that online. Once you get that in, you can transform it, use ampicillin selection, which is on this plasmid, and you can find one that is not fluorescent, which should contain your guide RNA instead of the GFP. Okay, so now you've got that, you can get the plasmid, you can overnight it, you mini prep it, and you get a linearized single guide expression cassette after an eco-RV digest. Okay, so that's your guide RNA is done. So you've got your linearized Cas9, you've got your linearized guide RNA. You want to bring these 2 together inside the cell. So you transform them into the cell, and then using gap repair, the guide RNA will slot into our Cas9 expression vector. So this one plasmid, which will be held inside the cell, will have your nuclease as well as your target. And like we said earlier, that would be enough to do some simple knockouts or whatever, generate some indels. But there's also the third part. Generally, we want to use another part, which is the donor DNA, which will give us a template for what we want to add in or what we want to precisely fix. And so for the donor DNA, it'll be, like I mentioned earlier, these recombination areas, which you can generally get through PCR, or there are some already designed for model organisms, easy to find. You can make this construct through PCR, through whatever cloning method that you have. In the toolkit that I described, the LS Lab toolkit, there is some plasmids already part of it, which allow for markerless integration, which means that there would be nothing inside, there would be no gene inside, except, and it uses the Ura homology. So it goes where the uracil would have been, and it finds homology arms, and instead it integrates a GFP, so you can see it. So yeah, so those are your 3 elements. You've got the nuclease, you've got the guide RNA, you've got your donor DNA, and then in the end, you transform it into yeast using whatever high efficiency yeast protocol that you use in your lab. We just use chemical competency of yeast, and then you add in each of your elements at specified levels. It's 100 nanograms of the Cas9, 200 nanograms of the guide RNA, and then over 2 micrograms of the PCR amplified donor, so your donor part. Or you can use the markerless integration cassette. But so you get these 3 things in, and you can do it in one transformation. It's really quick. And then what happens inside the cell is you should have this plasmid that includes your Cas9 and your guide RNA, and then it should make the edits inside the cell.
Yeah, so now we have everything. And let's talk a bit about the organism specificity of the CRISPR systems. We're going to talk about E. coli, yeast, plants, and mammalian cells because they're the most commonly used ones. So for E. coli efficiency, CRISPR Cas9 is not used that much, to be honest, because there are lots of other tools available for E. coli. It's really easy to work with. It's a model organism, grows fast, everything's perfect. But for more complicated organisms and eukaryotes, such as yeast, plants, and mammalian cells, because there are not as many tools available, CRISPR actually made a huge difference in terms of gene editing. It made gene editing way easier, very prevalent, and more efficient. So efficiency that I'm talking about here is more of an umbrella term for the overall process, because we can talk about the cutting efficiency, we can talk about the repair efficiency, knockout, knock-ins, activation, silencing. So it depends a lot on your application of CRISPR and which system you're going for. So overall, CRISPR is highly efficient in all of the organisms. It's only in plants, it's written moderate because everything that you do in plants is a bit moderate. It's a bit hard to work with them. So efficiency also depends on the length of the homology arms. So the bits that are matching to your matching to your genome that you want to edit. So for E. coli, it's around 50 to 300 base pairs. In yeast, it's anything more than 40 base pairs, and plants and mammalian cells are obviously a bit longer. And the reason why yeast is has the smallest homology arms is because yeast's repair mechanism is homology directed repair. So it's the prevalent mechanism. So it's really efficient. And to deliver, the efficiency also depends on your method of delivery. For E. coli, electroporation and chemical competence are the most commonly used ones, and the same is true for the yeast. For plants, there are two methods that are prevalent, and they are called agrobacterium transformation and biolistics. And for mammalian cells, electroporation and viral delivery are the most commonly used ones.
And just some last experimental tips that we haven't included on the slides, but it's one of those things that you kind of learn along the way, is that having the nuclease, the Cas protein, can be very toxic to your cell. So that's one of the hurdles of using Cas protein. Make sure that you're expressing it at a level that is not too toxic to your cells. And the second thing is to make sure in the cells that you're using, in the organism that you're using, what kind of repair mechanism is there and if that is compatible with CRISPR. Some strains already have like deletions in certain repair systems to make CRISPR easier. So those are two last things that I would recommend when choosing. All right, that's it for the experimental bits.